Rapids or Ann Arbor to the Medical Devices Forum. We've got a great bunch of folks here. As you can see in the pictures, we've got 3 folks in Ann Arbor. We have our moderator there and 2 of our company speakers here in Grand Rapids. Now, I'm not going to be a part of this. I'll be off to the side. We have 2 of our company speakers and our non-company speakers. So we have 2 sites that are connected in Grand Rapids and in Ann Arbor. We will be taping this so we can go back and edit it and put a smaller version of it on our website at some point in time out in the future. Next slide. As you can see, we're a little bit off of our timeline, but I don't think that anyone is in a hurry necessarily to leave. So we'll give just a couple of remarks. Our moderator will give an introduction to what's going on in Michigan. Then each of our 5 company speakers will speak for a little bit. We'll have the moderator will lead the discussion between our 2 sites, and then we'll have time for Q&A from both Ann Arbor and Grand Rapids. I think, and someone needs to look at the bottom of one of the computer screens, we're broadcasting this to Michigan Tech in Northern Michigan University, and I think they'll be writing in some comments or texting them to my phone number, 248-310-1027. And that way we'll make sure that their questions get answered also. Okay, go to the next slide, please. So before we get started, we want to thank some folks that made this possible. Number 1, Convene Web. Company's name is Creative System Dynamics. They're our technology backbone connecting all of us, and they put up with a lot. We're trying to work with me. So Gail, Neil, Mary, thank you guys. It's still an experiment, but appreciate it. Also, Logic Solutions of Ann Arbor. They've been our web host provider for a good long while now, and I think everyone that's looked at the new website can see some of the results of the work. They have been supporting behind the scenes along with our team, so they've been great. I want to thank our site hosts. In Ann Arbor, we're present at Menlo Innovations, and here in Grand Rapids, we're at GR Current, so thank both of them also. We're going to switch now to, I believe, the video is up next.
So the work we've done in our lab shows in many different areas that scaffolds, provide enormous guidance to help cells create new tissues. And we've published in proceedings of the National Academy of Sciences and other places the fact that in the spinal cord area that the combination of cells and scaffold clearly outperforms cells alone in getting much better performance. The polymer that we used and that in vivo is using are lactic glycolic acid copolymers to help support spinal cord repair. And the great thing about that polymer is not only is it FDA approved, but ultimately it breaks down into water and carbon dioxide. So it's very, very harmless in the body. It's been used in sutures. It's been used in various types of microspheres for drug delivery and other applications. So it's an incredibly safe material. That's why we use it. In the early 2000s and after that, we did a lot of studies where we showed that we could successfully treat rodents that had spinal cord injury using our approach. Recently, in vivo, published, and I was fortunate to be an author on that too, the fact that you could now use this on primates. In fact, that work won the Apple Award, which is a very, very prestigious award for spinal cord research. And the next step, we're doing more primates and now hopefully going to be going into humans in the near future. I had the pleasure of giving a lecture at the Sloan School in the mid-2000s, and I was one of the people who went to the lecture was actually Frank Reynolds, and he came up to me afterwards and asked about the possibility of licensing some of our patents to start in vivo therapeutics, which he did, and I've had the pleasure of working with Frank and in vivo since its inception, and I've been on the scientific advisory board and since that time and still today, and it's been very exciting for me to watch the progress that Frank and the in vivo team have made, now moving the early work that we started in small animals to now primates, and I look forward to humans. So it's been very exciting.
So I picked that video at the suggestion of Andrew Meadow, our moderator, because it's a perfect sort of example of what we want to talk about here today. Science, engineering, medicine, soft, squishy stuff, hard stuff, brain power, all coming together to form new products, new technologies which didn't exist before. So I think that as our panel starts talking, you'll hear more and more about that. So I'm going to just really briefly introduce Andrew Meadow. Everyone has at their place 4 pages that give the bios of all the speakers, so we're not going to give all of them. I'll let you read about Andrew. Andrew is Director of Biotech and Medical Device Ventures at MEDC. He has a long history from the East Coast as well as here in the Midwest of working in the medical field, both as an entrepreneur and in all kinds of other very involved positions of starting companies. I'm just going to leave it at that. You can read it. So, Andrew, I'll let you go ahead. If you can see on the screen the list of speakers that we're going to have. We're going to take them in alphabetical order. So if you don't know where you fall, that's the order you'll be presenting in. And the presentations are loaded in that order. So go ahead and pull up Andrew's slides, please. And Andrew, take it away.
Okay. We actually trying to see if we can bring up the slides here. Great. First of all, let me say that I'm the only member of this panel discussion that is not a doctor. So hopefully my 5 esteemed colleagues on this panel will take it easy on me in terms of the responses to questions. But first, thank you, Milton, for sponsoring this event and being the driver behind it. We're going to be focusing on 2 fundamental areas tonight. Rather than trying to boil the ocean about all the challenges and all the innovations that are taking place in medical devices, we are going to focus on 2 particular segments. One, personalized medicine, and the second, focusing on neurostimulation and neuromodulation technologies taking place. If we can go to the next slide now, please. Great. So, first of all, let's talk about what is personalized medicine and how do we define the next innovation frontier for medical technology. I mean, despite the economic and regulatory reimbursement challenges facing medical technology companies over the last 5 years, both neurostimulation as well as personalized medicine has seen exponential growth, not since just the last 5 years, but I would even argue going back to as far as 2010. In terms of the cost efficiencies these treatment opportunities have been able to provide early on in the treatment of some very rare and some very debilitating diseases, they've been able to identify specific solutions for patients and mitigate the long-term expenditures that reimbursement companies as well as hospitals have been allocating to the care of these patients. There are starting to become much stronger reimbursement and formulary codings for these treatment modalities, and as a result, over time, personalized medicine has seen a tremendous drop in cost. But, you know, it's somewhat, you know, a tip of the iceberg because those costs are, as we'll see, quite high nonetheless. Also, from a manufacturing standpoint, replacement sales for personalized medicine, particularly in the device and diagnostics arena, has seen a tremendous increase in revenues and impacted the bottom line positively for many of the players in the market itself, and also have provided safe and long-term therapies for a large, diversified and aging population. But most importantly, what I think is the most critical component of this is that there is limited competition in the space. This is still an incredibly new area, and every single day I'm reading about new innovation that's taking place in this space, particularly here in the state of Michigan. So I began to think about how do we define personalized medicine. If we can go to the next slide, please. And it is a very difficult thing. It was very difficult for me to define. And so I reached out to some of my former professors at Case Western Reserve and at the Cleveland Clinic to kind of tap into their opinion and insight as to how they define personalized medicine. But the most cohesive opinion that I found came from Price Waterhouse, so hopefully you won't discount that too much. But, you know, the segment of medical technology market that is dedicated to creating products and services that directly or indirectly leverage the science of genomics and proteomics, and that capitalize on the trends towards wellness and consumerism in order to facilitate tailored approaches to prevention and care. I mean, for me, I see it more as, in one area, advancing the sequencing of the human genome, which goes as far back as 2000, even 1999. The other component of this is achieving a greater understanding of the underlying mechanisms that are fundamentally at the root cause of debilitating diseases. Being able to take a treatment modality down to the cellular level redefines the way doctors and clinicians can define and treat debilitating diseases, and that to me I find to be incredibly compelling long term. And then, to give a softball over to Dr. Handique, the number of cancer drugs currently in development are being combined with companion diagnostic kits to help create treatment modalities that yield better outcomes. And I think that collaboration from R&D and diagnostics is definitely going to set a platform for better outcomes long term for these patients, and most importantly, improve the quality of their life and the types of treatments that they're getting. And then ultimately the ramifications with the Affordable Care Act. As companies are going to be judged by, and particularly healthcare service providers are going to be judged by the quality of outcomes in terms of the care that they're giving their patients, being able to treat patients early on in the diagnosis of a disease and at a very personal cellular or proteomic level is going to increase outcomes and reduce the amount of care that these patients are going to have to receive over an extended period of time. And if we can go to the next slide, Milton, please. And then I also wanted to set the stage by how we're going to be talking about neurostimulation and modulation devices. First of all, neurostim has been gaining acceptance as a therapeutic alternative for chronic conditions for about 5 years now. There are significant big pocket players who are in this space, and they have found uses in the management of movement disorders, including Parkinson's and epilepsy, that have fundamentally redefined treatment protocols for those debilitating diseases. They've been able to approach those disease states in a way that biopharmaceuticals have not been able to in the past. The other thing is that we're now looking at new implantable devices for deep brain stimulation, gastric nerve stimulation, sacral nerve stimulation, vagus nerve, and spinal cord stimulation. And actually, as we begin to look at neurostimulation as an overall market segment, 50% of that market is really dedicated to spinal cord stimulation, which we'll talk about a little bit more further. But I think the greatest impact that we're seeing with neurostimulation and modulation devices is the fact that we're looking at a market that just last year was looking at about $1.9 billion in total revenue. And in approximately 5 years, we're looking at an industry that, conservatively speaking, is going to be generating approximately $6.5 billion, or 17.9% CAGR over that same period of time. But one of the biggest challenges that is facing this particular industry is market acceptance and gaining reimbursement for this space. As of tonight, I have seen very little formulary coding for neurostim or neuromodulation, and that creates a challenge for both the consumer as well as the care provider and what they can and cannot do. So these are going to be some of the topics that we're going to drill into tonight. And with that, Milton, I believe you had in mind going to, was it David next?
Tim.
Is that correct? I'm sorry?
It's going to be Tim. It's me, Tim
Okay.
Fischell.
I'm sorry, Tim. I apologize.
No problem. So I've been asked to speak very briefly about a company that I founded about 2 years ago called Ablative Solutions. This is a company founded in 2011, and our first target is medically refractory hypertension in a relatively large space now that's called renal denervation. We have a novel, if not unique, approach to, rather than using radiofrequency or burning type devices to burn the inside of the renal arteries, and I'll get to this in a second, we are a chemical renal denervation company using ethanol, which I'll get to in a second. We've actually completed our first-in-man experience. Haven't completed the first-in-man, but we've done our first 10 patients, and with some very interesting and encouraging initial results. The company, although it's only 2 years old, we've already filed 12 patent applications with several notices and So we're very active in protecting the IP of what we're doing. This was the leadership of our company. We're a very virtual company in that we only have really two people working, getting paychecks from the company. That's myself and the president, Dr. Gazarosian, who's in Menlo Park, California, actually. We did raise $5.3 million in our A round in 2012, which included investment by BioStar and Michigan Accelerator, and we're just closed last week about a $9.5 million B round. We can go to the next slide. This is just a brief bio of myself, so you can look at that on the paper. We're not going to go over that. Next slide. I will say that I've been involved in medical devices for about 20 years now, especially with my father, Robert Fischell, who's a very well-known, won the TED Award and some Inventor of the Year in the United States. You could go through his 200 patents. I have about 85 issued patents, and we founded about 10 companies, including 2 in neurostim neuromodulation, including NeuroPace, a closed-loop brain stimulator for epilepsy that was approved about 2 months ago, and eNeura, a transcranial magnetic stimulation for migraine relief, which is an amazing device, which was approved by the FDA about 4 weeks ago. We had 2 approvals from the FDA. But that's not what I'm talking about tonight. I'm talking about this other company, Ablative Solutions, and our goal is to treat hypertension, to create a catheter that can go in and kill the sympathetic nerve fibers going from your brain to your kidney to reduce renin release, angiotensin II production, and aldosterone, and thereby reduce blood pressure. It has multiple other effects that are really, I can't go into depth because of the time, but I will say hypertension, as you all may know, is one of the biggest medical problems in the world. 1.2 billion people with hypertension, about 15% of those with refractory hypertension. That's 3 or more medications and a blood pressure over 160. So this is a very severe problem. There are a lot of people with it, and it's one of the biggest costs to healthcare is the cardiovascular manifestations that arise from the risk factor hypertension causing stroke, heart attack, and death. Next slide. So the interesting thing about renal denervation, when you knock this circuit out from the brain to the kidney and the afferent and the efferent nerves going from brain to kidney and kidney to brain, you not only reduce blood pressure substantially, you also, it has been, not surprisingly perhaps, a dramatic effect on congestive heart failure, left ventricular hypertrophy, diastolic dysfunction, chronic kidney disease, type 2 diabetes, glucose intolerance, obesity, atrial fibrillation recurrence after ablation, and sleep apnea, central sleep apnea. It has published papers in all of these scientifically proven benefits when you denervate the kidney. Pretty amazing. Next slide. So it's not just hypertension. There are widespread effects in the body. This is our unique approach, what we have now trademarked as perivascular renal denervation, or PVRD, where rather than burning from the inside of the artery with a radiofrequency catheter or ultrasound to burn through the intima and the medial layer and destroy the artery to get to the nerves which are in the outer layer, and you can see what we do is we center a device with tubes and 0.008 of an inch nitinol needles that go through the wall, and then we inject 8 drops, 0.3 mLs of ethanol, which is highly neurotoxic, spreads in the fat tissue around the artery, kills the nerves in about 30 seconds, and is painless instead of extremely painful when you burn through the wall. So it is controlled perivascular targeting to where the nerves are, which is in the outer layer, without damaging the artery, efficient simultaneously delivering and destroying the nerves in the adventitial layer, self-limited spread due to the very small volume that we're using. There's no obstruction of blood flow, no injury to the artery, and no capital equipment cost. These boxes that where you hook up the RF can cost $30,000 or $40,000, and now there are like 15 companies trying to do this, and they're going to be, who's going to buy the 4th box? Next slide. We have no box. So when we look at our cells, chemical neurolysis by Ablative Solutions, compared to, for example, the first generation RF system made by a company called Ardian, bought by Medtronic after they did about 100 patients, bought for $1.2 billion after doing 100 patients and being 4 years away from U.S. approval. So that's how much excitement there is in this space. It is perhaps the biggest medical device space in the history of medical devices. That is the treatment of hypertension. So, but compared to first generation RF, We can get the deep nerves. They can't. We cause minimal injury to the artery. We do this in 90 seconds per artery is the entire procedure time once you put the device into the artery. Very little contrast, essentially painless, no capital equipment cost, easy to navigate, almost no anatomical limitations rather than having to burn long segments. We can deal with almost any anatomy. We do not cause stenoses of the artery, unlike the burning. We do not cause clots on the inside of the artery. We can predict and titrate the dose, and we even have patents now on sensing the nerve activity in the sympathetic nerves at the same time that you ablate. So we have many advantages versus certainly the first generation Simplicity device by Medtronic, which is finishing its U.S. trial. It's probably about a year and a half away from approval. Second generation, where they've been so clever as to put 4 electrodes on a balloon and burn 4 places at once. That's the shocking, brilliant innovation to the second generation that still burns and hurts, but it's quicker. And then ultrasound, which also requires an expensive box and burns and hurts and damages the artery. So we believe we have a way of knocking out 80 or 90% of the nerves instead of 30%, in 90 seconds instead of 20 minutes, and with no pain. So we are very optimistic about the future of ablative solutions. I think next slide, last slide, I think. So renal denervation is a very promising means to reduce blood pressure. I'm not showing all the data. Obviously, there's excitement about this because it works. So I'm not going into all the published data. There's about 100 published papers now about renal denervation, which you can look up. And we think there are many potential applications of both the device, not only in hypertension and renal denervation, but in other parts of the body where we can deliver drugs or chemicals to ablate tissue or other therapeutic agents. And we believe that chemical renal denervation may overcome many of the limitations of both first and second generation radiofrequency ultrasound systems. And our early human data is encouraging. Thank you.
Thank you, Tim. And then I guess after that it will be Kalyan. We're going in that alphabetical order. Okay.
Hello, everyone. I'm Kalyan, Handique, CEO and President of DeNovo Sciences. We are a young cancer diagnostic startup based in Plymouth, Michigan. We started in 2011 and have been really going through the paces of developing the product to be launched very soon in the research market. So what we are building is a next generation microfluidic circulating tumor cell detection system directly from blood. And as you go through my slides, you'll realize, why it's important, and the technology that you're developing is why it's so relevant to personalized medicine. Next slide, brief bio. I joined DeNovo Sciences in 2011. August of 2011. Prior to that, I was with Becton Dickinson because they acquired a company I started called Handique in 2009. So 15 months into BD, I knew that I had to get back to the startup, and then, was figuring out where to go. And then DeNovo Sciences was one of the companies I was mentoring, and they really started picking up steam. They won one of the business plan competition through GLEQ, the best business plan, and that's what really validated the business that DeNovo Sciences team was going forward. And I stepped in, and then no pay for a couple of months, gave myself a deadline. Okay, if I can't raise money in the next 4 to 5 months, we'll go back to some other things to do. So fortunate enough to win Excel Michigan, the grand prize win, $500,000, was able to leverage that and get folks like Walt, who's in the audience here, and a few other angel investors to put in all in all about $3.3 million into the company. We've been very capital efficient, and then took the went through the development of the system, and then would be launching the full automated platform in a few months. A lot of things have happened in the last 2.5 years. Fortunate to be in Michigan with a lot of talent and a lot of support from the different elements of the community. So what is the problem that we are trying to solve? We are essentially trying to place ourselves in a nice position to be able to impact cancer, not only in the detection side, but hopefully in the therapy side eventually in the future. Why is it important? Cancer is very soon going to replace cardiovascular as the leading causes of death in the United States. And then it's also probably one of the leading causes of death, for the whole world. In U.S. alone, you have about 600,000 people die every year because of cancer. Every year you are diagnosed. I think it's about 1.6 million new cases of cancer every year. And these deaths happen not because you have cancer, but because your cancer has metastasized from a primary tumor to other parts of the body. Once that happens, it is pretty much going downhill. You're just trying to delay the inevitable death. You're just trying different cocktails to give you more life to the patient. And, the burden, economic burden is so huge, $200 billion, both direct and indirect. And some studies said that if you can reduce the debt by 1%, you'll probably save U.S. $500 billion. $500 million, just 1% reduction in debt. So currently the gold standard for detecting cancer is biopsies. You probably use some technology like CT scan, MRI to see if there is some suspected mass in your body, and then you have to do biopsy to do definitive diagnosis of what's really happening. And it's very hard to do biopsies on patients that have metastasized because they are already very weak, frail, and it's sometimes you cannot even get to the site of where it's metastasized. For example, prostate very often metastasizes to the bone, and you don't have any sample. So, it's been a big problem, not only for, treating those patients, but also it's been a big problem in the research side. You can't even get, samples to do your research on new drugs. How can you really solve the problem? That's where, we think that we can provide a solution to this immensely difficult problem, a blood-based liquid biopsy. Can you go to the next slide? Okay. So what we are focusing on is called circulating tumor cells. These are the cells that are, shed from the primary tumor, and they go through the bloodstream and often also through the lymph stream to other parts of the body to create secondary, metastasis, secondary tumors. So why not do a blood draw, fish out these cells, and do all the biopsy-like tests on these CTCs? Again, it's a nice concept, but it's not an easy problem. You have only a few cells in milliliters of blood in a background of billions of other cells. So this is a real needle in a haystack problem. So it's technologically one of the most challenging tasks this world faces in this century. In fact, if you read the publication in the last 5 to 10 years, this is like the holy grail of healthcare. If you can detect CTCs and allow biopsy-like tests to be done on the CTCs, you can probably cure cancer. And that's where, the personalized medicine comes into play is, when the cancer metastasizes, it can manifest in lots of different forms. So if I say some person has, late-stage prostate cancer, there is no one cure for it. There are so many different manifestations. You really have to figure out what's happening at that point of time with the patient so that you can give the right drug, so that they can work on the right target within the cell. So we think that we have a solution for this really difficult problem. Next slide. And to make it easy, we are building a complete system around, doing that biopsy-like test on CTCs. We have a microfluidic slide that allows you to fish out those really difficult to, capture cells. This one has 56,400 cell capture chambers. So you run blood through it. 2 ml of blood takes 6 minutes. And it's able to capture those rare cells in this device. And then you have the system the JETA, which is a system allows you to do automate the biopsy-like test on the same slide. And the Vanguard looks through all the chambers and gives you the result in the same machine. And we have not only developed the technology to capture CTCs, we have been pretty much going through the list of tests that can be done on those cells, whether it's immunochemistry, PCR, looking at mutations in those cells, FISH analysis, looking at, again, actionable mutations on the cell, and even grow the cell in the same chip. So we think that we have a solution that can revolutionize not only early detection of cancer, but eventually also start, tailoring, drugs per patient. And then so that's where, we think we'll have a system that pharmas would love to start, marrying up their new drugs with a specific test. Next slide. So how are you unique? We have the only system available now that can do this very difficult, extraction and all the different biopsy, like whether it's cellular, genomic, analysis in the same device. Because these cells are so rare, you cannot just, cobble together different, machines from different companies to get the results. You really need one integrated solution. That's what we provide. Thank you.
Thank you, Kalyan. Brent?
Hello, my name is Brent Nowak. I am the founder and CTO of a company called Synosteo, where we're developing a new technology around bone regeneration. Next slide, please. The vision for our company is that we want to provide personalized medicine, and going back to Andrew's previous definition, I think that definition is a little bit narrow, and especially in the case of trauma, you can personalize medicine on an individual patient basis as well as the healing stages they're in. So I broadly then, for the sake of the discussion, expand that definition of personalized medicine. It's not just genomics or proteomics, but in this case, we're able to help the body heal the bone naturally at a much higher rate with better outcomes and as much as 1/2 the time, and a reduction of the related costs that go along with that. Go ahead, next slide. Now, when I was talking to Andrew about who we were talking to today, part of the audience was going to be students and those people who might be involved in startup companies or innovation. And the reason I put my background up in that fashion is that, I got my first degree from the University of Illinois in mechanical engineering in the field of heat transfer and fluid dynamics. And then I worked in industry for a while and went back and got my master's degree and started studying robotics and biomechanics. Worked at NASA for a while, then went back and got my PhD in robotics. So the message I wanted to give in my little story here is that there is no direct path to medical device development or innovation or entrepreneurship. You take your background and you follow your passion, and if you're creative or you team with the right people, there's a lot of ways to get to where we are today, and you just don't have to have a PhD, nor do you have to have an MD to be an innovator in this field. And again, you can read all the stuff I've been doing in the last 5 years, 6 years. Next slide, please. So bone regeneration, the market, this data is a little bit, this is dated, and I apologize to that. That goes back to 2002. But it's a $2.72 billion market. You can see some of the pictures up there, the post-traumatic world, whether you're a soldier, as we know the last 2 wars we've had in Afghanistan and Iraq, or you're in a car accident. There's numerous congenital disorders, birth defects is another way to say it, tumor reconstruction, but also you can get into elective surgery in the snoring situation there, which also I guess could be a sleep apnea issue. So it's a very large market. These numbers come just from the United States alone. Next slide, please. The challenge is that what I want to address today when I talk about bone regeneration is a process known as distraction osteogenesis. And for the audience, you may or may not know, but distraction osteogenesis is a technique developed by a surgeon, Dr. Elizarov, out of Russia, back in the 40s, I think it was, where basically there's an osteotomy site. The surgeon does a surgery and basically cuts the bone, and you put some brackets on it, and you slowly stretch it out. So in the left-hand picture there, you see somebody who's doing a craniomaxillofacial process where they're actually modifying the bone structure of the face itself. The way the Elizarov method works is there's a little incremental screw. Basically, you turn it 1 to 4 times a day, and you distract anywhere from 1 to 2 millimeters a day. And it's the same distraction process used worldwide. And so it's every time you have to turn that screw, you get to induce a little bit of pain on yourself. And if you have a child or a loved one, and you bring them home and you have to do that every day, it's quite a tragedy if nothing else. But on the far right, you can see some of the problems we have. There's a very complex distraction osteogenesis process where it's a 3-dimensional, 6-dimensional actually problem that they're trying to solve by growing new bone. I can't tell you what the cause of that particular one is. I have some wonderful slides if you want to some of our soldiers are going through today. So what we're addressing in our solution is dealing with the issues of the pain, the cost, and patient noncompliance. Next slide, please. What our solution does, and this was an early graphic, one of my co-inventors is Dr. Jonathan Junt. He's a cranial maxillofacial surgeon, and so one of the first things he wanted to address was the mandible. So what you see there is a conceptual drawing of our solution. There would be a cut between the bracket and that rod that goes on there. And what we have is we have a closed-loop adaptive system that senses the bone healing within the human body in real time. So that's showing a subcutaneous installation. Our first trials are going to be, animal trials will be a transcutaneous on a large bone, specifically a boar goat. And what we're doing is we're sensing how well the body is healing the bone at any particular healing stage, whether it's in the early latency stages or later when we're starting to get to the consolidation phases. With that information, we're able to take that information and send it through a Bluetooth device, and the surgeon or the clinician or physician can see how well you're healing and can make some decisions on how to modify that. So in this process, in this closed-loop process, we do continuous distraction. That continuous distraction rate may change depending on how the physician wants to increase or decrease that rate, depending on how you're healing. So you may be healing differently than that gentleman here, which may be healing than a young lady in the back, or somebody who's a diabetic, or somebody who's a smoker. So going back to the personalized medicine terminology, on an individual basis, we can now respond to how you heal at any particular stage and optimize that healing process. Next slide. So the benefits of this is we expect to get significantly higher, like I said, as much as 2 times quicker healing rates. We can overcome the issues of patient noncompliance since it's totally closed-loop control. This process will be, the patient will not, or the patient's loved ones won't have to interact with the device at all. We can, one of the things I didn't talk about is that when you have this kind of process going on, the way that the physician studies or determines how well you're healing is either they palpitate that injury space, which is painful, or you go in for x-rays, sometimes as frequently as every 2 weeks. So in our process, since you can get the in vivo data real time, you can eliminate the need for going for radiation imaging. Ultimately, when this, since we're doing it for a shorter amount of time, you have less likelihood of infection. When it's a truly fully subcutaneous process, obviously you won't have any path for infection at all once after the surgical procedure is complete. Obviously, as I mentioned before, we got real-time monitoring for closed-loop control, and we'll be able to drastically improve the surgical outcomes. So the insurers and the practitioners will be able to grow the market as well. So what's unique about this is that whether you're using a Smith & Nephew fixation device or a Stryker fixation device or orthofixes, this technology can be applied to any of the existing devices that are used today. So what we truly have is a platform technology that can be used on any bone in the body and that can be used with any existing technologies that are available today. And when we look at our competition, whether you're talking about implantables or biologicals or pharmaceuticals or something they call scaffolding, this technology can be used in those applications as well, and still provide that same kind of information to the physicians. Next slide. So one of the other topics we were going to talk about today, and this is my little pitch here, is about medical device innovation. We're in a time in history today where, with the continued march of semiconductors, miniaturization, multi-core processors, better sensors, we're seeing more and more implantables in the field. One of my regrets is that I'm not 20 years younger, because we're going to see more medical device innovation, more implantables. We're going to see more medical devices on people, in people, communicating amongst yourself in the next 10 to 20 years. It's a very exciting time. So as I say in the bottom, I think we have a rich opportunity for individualized healing, or in quotation marks, personalized medicine in the medical device space. Thank you very much.
Brett, thank you very much.
Sure, Andrew.
Just so everybody knows, Brett left San Antonio, Texas, either on Saturday or Sunday to drive back from Texas to Michigan. And Brett hit the eye of the snowstorm. You got back today, is that correct, Brett?
Yeah, now it's about 3 hours ago I arrived in Grand Rapids.
So thank you very much for participating under those circumstances. Next, I have Dave Olson on here, who is, and one of the things that Kalyan left out in his little presentation is that the Michigan Venture Capital Association decided to do a promotional event this year where they came out with the all-stars of innovation in the state of Michigan. And both Kalyan and David received baseball card-sized accolades for their accomplishments. And but David is one of our returning successful entrepreneurs, and we're very glad that you're here, David. Thank you.
Thank you. Let me bring the mic up, I guess. Right. So I'll start with a little bit about my company, Swift Biosciences. Co-founded it with an inventor here in town. We're a biotechnology company. We're focused on enabling technologies for genomics and personalized medicine. I'll talk a little bit about what we make in a minute. We're a venture-backed company. We're a Series B stage company. Investors include venture funds that are listed on the screen, as well as prominent local individual investors, otherwise known as angels, at least one of which is in the room. We opened doors a year before Handy did. We're about 13 people today. We're based in Ann Arbor. And we're driving our value through partnerships, which we have with diagnostic company, and direct retail of our products. So, I got the next slide. A little bit about myself. They asked for a bio slide, so I put one in here. I'm not going to spend much time on it. I'm a molecular biologist by training. I think about how to manipulate DNA and work with enzymes. But I left the bench a long time ago. I've been involved in entrepreneuring for many years, and have worked in just about every field in the life sciences. I enjoy it all. Okay, so on the next slide, yeah, I think we all have our definitions of what personalized medicine is. I'll broaden it even more. I think we've had personalized medicine for a long time. For instance, using the example of cancer, if anyone's ever had cancer or our family members had cancer and the cancer was staged, that staging basically identified what type of therapy the patient is going to receive. So there's any number of diagnostic technologies that are used to identify what a patient needs. I think what we're talking about here is a new horizon of technologies that are being brought to bear. Traditionally, we have imaging, patient history, surgical findings, surgery for doing biopsies. If you have biopsies, you're going to get microscopic findings from the pathology department. You're going to get a report to the physician. You're going to get treated. What's happening, if I have the next slide, please. There we go. So what's happening in my field, and Handy's, and we're in the same field, the molecular diagnostics. And as Handy mentioned, that's really driving a tremendous amount of research. Importantly, drug development. It's giving an insight into what's going on at the molecular level, and new treatment options. We tend to think about, the industry tends to talk a lot about molecular diagnostics for cancer. Molecular diagnostics are being used for other diseases as well. The molecular diagnostics are the most common type of technology that's being applied to what we call companion diagnostics. That is to say, a drug which has a diagnostic that it is in tandem with, and only through the use of that diagnostic tool can you identify which patients are classified to be treated with that drug. So that's the field that we're in. And very briefly, if I have the next slide, please. Thanks. Okay, so Handy's very much in the business of identifying cells that are basically cannot be seen by a traditional biopsy. We're in the business of finding lesions in DNA that can't be seen using the available molecular technologies. Handy talked about using PCR as one of the diagnostic technologies. we specialize in an area of that of PCR. We also focus on sequencing and have technology to improve next-generation sequencing. Unlike my fellow speakers, I can't show you a prototype because what we have is actually a little bit of water in a tube, and I could show you that, but it's like this, only smaller. And it's what's in the water that counts. And basically, we sell wetware, reagents. These are consumables that are used in existing lab instruments. So Handy, for instance, is building an instrument that does something. There are other instruments out there that do different things. Those that do sequencing and PCR, we provide the liquid consumables for those, and those machines are able to do a better job of what they're intended to do, and in some cases are able to do things which they couldn't do before. I mean, that's very important from a medical perspective. We're able to provide the ability to do diagnostics on samples that were otherwise not available to physicians. And sequencing in cancer, as much as 20% of patient samples actually can't be sequenced. There's not enough material there. And there's been a big push in the field to provide greater information out of smaller samples. We excel at Swift in the ability to distinguish between the presence or absence of a mutation when there's very little there. And this technology is complementary with the types of things that Handy's working on, which is basically a new type of biopsy, grabbing cells that otherwise could not be seen, and then we can look inside those cells using the molecular technology. All of this leads to a world where you can understand more about a patient or an individual than is possible by looking at them with standard techniques. So I'm looking at the audience here. I can get a sense of how tall people are, how much they're squinting at me. Now we can actually begin to look at what your genetic profile is. In Handy's case, we can look at what's actually circulating in your blood and whether or not there are cancer cells there. And if there are cancer cells there, what types of mutations do they have? As a sort of lead into some of the ethical issues, these are all, we speak about these as unmitigated good things. And these technologies can be used to do amazing things and are being used to do amazing things that simply weren't possible. People who had diseases that the doctors had no idea what they were, and through sequencing they were able to recognize that the patient might have an odd disease that was otherwise not known, take a guess at what the medicine might be, and actually treat patients. There's some remarkable stories out there. But of course, sequencing can also be used to identify individuals and get a sense of what might happen later in your life. We know a lot about what your DNA says about what will happen in your life. So collecting that information, deciding what information a physician is going to use. So for instance, if we sequence the genome and we're only looking at a couple of cancer-related genes, which we want to find out whether or not we're going to treat or not treat, what do we do with the rest of the sequence that came along with that, right? That might be information that's, well, your insurance company certainly would like to know that because they do a better job of actuary work that way. But that's probably not what you want. And with more advanced technologies for collecting this sort of information, in just the same way that what happens in your life can be tracked by where your phone goes and what it connects to, we can start tracking things at a molecular level by looking at DNA. I think there's very exciting things going on with being able to monitor patients in real time for the bone repair that we just heard about, whether or not patients are being compliant with medication, whether or not patients are having difficulty sleeping or moving. And there's all kinds of exciting things going on. All of these, I think, could fall into the realm of a personalized or individualized care all of them also can be abused to look at what's going on with individuals that otherwise we would have expected a privacy barrier that had existed before and probably won't exist 10 years from now. So with that, I'll finish.
David, thank you very much. And Michael, Michael is a clinical ethicist from Spectrum Health, and perspective on personalized medicine relative to the other four panelists who are actually in executive and bringing products to market. So, Michael, if you can start, please.
Yeah, sure. Thanks, Andrew. So just start by saying thank you to Milton and Andrew for their hard work in putting together this forum. I'm going to go a little bit off script, partly because of time and partly in response to some of the other comments and stuff. Real briefly, my role at Spectrum Health is as an ethics consultant, primarily for issues related to bedside care, so patient care issues where you have some sort of ethical question or ethical dilemma that's come up. So that can range from some fairly standard questions about informed consent, surrogate decision-making, does a patient have decision-making capacity, a lot of end-of-life decisions, so when do we stop treatment, when is it appropriate to let somebody go, to all sorts of questions that arise because of a new technology or a new capability that we have. And for me, those are the really exciting questions. And so just to give you an example, Brent and I were talking earlier about these new cardiac devices that are being used more and more called ventricular assist devices. It's essentially a heart pump that is implanted in an individual with heart failure, and it's connected to an external part of the device, usually a control unit or a battery. And it's a big deal to go through this procedure, open heart surgery, obviously, life or death situation. Patients decide they want this done, and then some of them, at a certain point, 3 months, 6 months, a year later, decide that this is not what they really want. And so they come to the clinicians and they say, I want you to stop the device, turn it off. Well, of course, that's their heart stopping, and in most cases that individual will die in a couple of minutes, very quickly. So what do you do in a case like that? Well, if we had transplanted that patient, if we had put a human heart in them and they came to us and said, take the heart out, I didn't really want that, we would say, sorry, can't do that. It's in you, it's part of you, belongs to you now. On the other hand, if you had an individual who was on a ventilator and that device was assisting with their breathing, taking over their breathing function rather than their cardiac function, they said, turn off the ventilator. We do that every day. I mean, that would be not uncommon at all. So what category do you put this new device in? Is it something that belongs to you, it's part of you, we can't take it out or turn it off? Or is it more like this external device? And those sorts of questions, it's an interesting question, even, philosophically, theoretically. So that's the type of dilemma that we sometimes see at the bedside that comes up because of a new technology, and it's really my privilege and my role to be able to try to help the clinicians sort some of those questions out. Next slide, please. Okay, that's our moral admonition for the evening. That's actually from Thomas Aquinas. He said that's the first principle of practical reasoning. Avoid bad things, pursue what's good. And so what is the place of ethics in a conversation like this about new technology? I think one part of it, an important part of it, is identifying what are potential harms, what are these bad things that we should try to avoid. And in some cases, they're fairly obvious. So if you develop a new a heart stent, and in testing you find out that it actually causes strokes in your patients, then that's not a good thing. That's something you want to avoid. That's fairly straightforward. In other cases or situations, the types of harms are potentially more subtle. So when we get into the realm of personalized medicine, I think some of the challenges have to do with what do we do with all this new information that we now have, this genetic information about a person. So how could information be a bad thing? Well, interestingly, so if you look at some studies they've done on patients with terminal cancer, and you ask them, do you want to know your prognosis and your life expectancy? Consistently, in the studies, about 40% of patients will say no. I'd prefer not to know that. And for certain people, they don't want to live with this time frame hanging over their head. That information can be distressing in certain ways. Sorry for if you guys haven't been able to hear me here. So information can, in some cases, do harm. Next slide, please. So one example, this recent example that's been in the news, you guys probably know about the direct-to-consumer genetic testing company 23andMe. In late November, they received a very stern warning from the FDA that they are to cease and desist from their advertising and offering this $99 genetic testing kit, which would give consumers all this information about their genome and their predisposition toward various sorts of conditions. The FDA was concerned, first of all, that they hadn't established the reliability of their testing. But another part of their concern was, what are consumers going to do with that information? How are they actually going to use that? How do they interpret it? What does it mean when you say you have a 20% higher predisposition toward Alzheimer's? And their concern is that they might misuse some of that information. They might actually try to self-manage their own disease, their medications, what tests they should or shouldn't get, without the consultation of a professional, like a physician. Next slide. So when we get into personalized medicine, again, there are these interesting questions. What do we do with the information? One of the other, the last speaker referred to the issue of incidental findings. What do you do with the potentially significant health-related information that you find in a person's genome that you weren't looking for, the stuff you find by accident? Some of that could be clinically relevant to patients. They would want to know that. And so then you might say, well, then we have an obligation to tell them, even if they didn't ask for it. Some of it could fall into that possibly harmful category, where they have a 20% higher chance of pancreatic cancer, and they're going to live the rest of their life fearing that they might develop cancer, or they might go out and get a series of tests that they really didn't need, biopsies, etc. So incidental findings is one of these interesting, challenging questions. Another one is, if we're able to differentiate between patients who respond well to a particular drug and patients who respond poorly based on their genetic profile, can and should we use that information to ration? Now, healthcare rationing is a hot issue. A lot of people, the question of whether we should use cost effectiveness at all when we make treatment decisions is a hot, contentious topic. But I think personalized medicine is going to make that even more pressing, because suppose you have somebody who has lung cancer. With the standard therapy, their prognosis ranges from 6 months to 4 years, say. In the past, knowing nothing more about the patient, they could say, I want to go for it. Course of therapy costs, say, $70,000, so not cheap. Well, that person might be the person who lives for 4 years, and then you'd say, well, that's not a bad return on investment. That's a good way to spend healthcare dollars. $70,000 for 4 years, not too bad. $70,000 for 6 months, that's starting to get a little questionable. Okay. With personalized medicine, we might now be able to differentiate between the patient who's going to be the 6-month patient and the 4-month patient. Should we use that information to make cost-effectiveness rationing decisions? So I think that's a set of questions that personalized medicine is going to make even more pressing for us, even more challenging, especially as, looking into the future with the economic challenges our country faces and the healthcare system in general. So I will leave it there and look forward to the Q&A.
Thank you very much, Michael. And as a follow-up on something Michael said about incidental findings, the Presidential Committee for the Study of Bioethical Issues urged doctors, and this is something that was determined a few weeks ago, that urged doctors to come up with a set of guidelines themselves for dealing with incidental findings that are bound to come up when they're dealing with the patient's genome, and that genome receives the level of scrutiny that we're talking about. And so a lot of the rules and regulations, whether it be on the point of care or whether it be at the level of the FDA, a lot of the rules and regulations governing personalized medicine are really being developed on the fly, unfortunately. So for the sake of time, I'm going to curtail a lot of my questions so that you all have plenty of time and opportunity to ask the panelists some questions. But I am interested in asking a general question to Tim, to Kalyan, to Brent, and also David about this challenge of what is personalized medicine, and the fact that 5 panelists come up with arguably 5 different definitions of personalized medicine. You can just imagine how difficult it must be for the FDA to do that as well, and to monitor and regulate this industry. So I'm curious what challenges that you all have faced as you've been going through your development life cycle, and what changes you would like to see or hope to see in the near future. And maybe, Kalyan, you can start off.
Well, the concept of personalized medicine is not something new. It pretty much took hold within the scientific community as soon as the human genome was sequenced, and there was a lot of hype around that time. And then we are only beginning to see some of the technologies become products. So it took us a while, in almost, I would say, 20, 30 years to get to where we are. Some of these things have been taught about almost 20, 25 years back. So it takes time. So there's a natural selection of good products process over that time period. So only the good things will survive, because the bad things. That was true on me when it came out. I knew that's no way that's going to survive the FDA environment, because I lived in the diagnostic world, and I just couldn't figure out how could something like that stick around for too long. I think they stuck around longer than I thought they would. So if you ask people in the field that really knows the details of what it takes to make a good, reliable diagnostics, they will know that some of those will go away. So I would just leave it out for natural selection to have the good technology survive. I think it's good for, the healthcare system in all. FDA is a great balancing, does a great balancing act. The whole world outside actually looks at US FDA products as the gold standard. So, it makes things slow, but I think it's all going to be good for healthcare, for the world, in fact.
Tim, do you have any thoughts on this?
Yeah, as a practicing physician for 30 years, I'm going to be a little bit counter. The ethicist is supposed to be the counter technology person, but I'm going to be a counter technology person here a little bit. I'm going to throw out a challenge and say we're so focused on the microscopic genomic, one mutation in one cell on one thing, so we can maybe figure out that this cancer drug might improve the outcome by 5% and spend another $100,000 on one patient. We're really missing the boat, and the boat has left the dock here, folks. Anyone who's trying to develop technologies that are going to try to do these things, it's over, because here are the problems that we're not facing. How about just drawing your LDL, your low-density lipoprotein, and treating high cholesterol with a generic drug? Now, that's cost-effective medicine. It's probably 1,000-fold the cost-effective of anything we heard about tonight. Or treating hypertension, of course. Population-based health care cost reduction and improvement in health, or obesity and eating. So obesity, eating, fitness, lipids, hypertension, smoking. These are systematic, population-based, massive problems that are driving the healthcare costs. And I think I'm just going to be counter and say, finding that this person has a cell, or better yet, a gene that predicts that their breast cancer risk, this woman's breast cancer risk, is 17% higher. It's 21% in her lifetime instead of 15%. And what does that mean? And we're going to spend, $20 billion testing everyone's genome so we can find out that this person has a 5% increased risk of some form of cancer and a 4% less risk of cervical cancer. And okay, so stop smoking, treat your blood pressure, exercise, lose weight, eat right, and don't smoke. I mean, that's literally probably 1,000-fold the cost savings of anything we've heard about on these testing. We're missing the big picture of the forest for the trees.
Yeah, Tim, that applies to simple bodily conditions. For something like cancer, where they have been trying to unravel the mysteries, your simplistic theory won't work.
Well, before we drill down to that, I'd also love to bring in Dave in this, who's spent your career in manipulating DNA and working with enzymes. What is your take on Tim's approach to personalized medicine?
Well, I think, Tim, you're still welcome in Ann Arbor.
No, I think the technology you guys are doing is great. I'm just saying we're in a day and age where 25% of the hospital is going to go out of business. Our hospital is looking at $190 million of Medicare cuts in the next 10 years, and possibly forcing bankruptcy. And so we're in a place where we have to start treating populations and reducing costs. And I think it's all great to be able to individualize cancer treatment. These are noble goals, and they're challenging goals, and these are great technologies. I just think they're going to be very challenging in the next 10 years.
I would agree with that for sure. And in fact, there have been studies where someone volunteered their genome to be analyzed and asked, okay, you've analyzed my genome, now give me a prognosis. And the prognosis was, well, you should exercise regularly, eat right, and don't smoke. So I think that would be an example of an unnecessary diagnostic. There's really no point in most sequencing the genome to look for things just like whole body scans, X-rays, that sort of thing, tend to be expensive. They find things that patients worry about, but they generally don't catch anything that's meaningful. On the other hand, there are effective drugs for some types of cancer that are highly effective, but only in a subpopulation of patients. And then you're left with if we're going to treat patients for these cancers, and we have a drug which works well, but will be actually maybe ineffective and even harmful in patients in one category, then we're left with the it's beneficial and I think ethically valid to differentiate between those patients. I have to say that the question of the 6 months versus 4 years is a thorny one that I'll try to avoid entirely by focusing on what's in the water. But I think that trying to deal with the rationing of healthcare and the cost of healthcare is a problem that's certainly beyond the scope of what we're doing from a molecular analysis. How that molecular analysis is used and when that molecular analysis is used, I think will have a big impact on whether or not it adds to the problem or helps simplify and reduce the problem.
That's great. Thank you, David. I want to make sure with the limited time that we have left, and again, my apologies for starting later than expected, that the audiences in Grand Rapids as well as here in Ann Arbor have an opportunity to ask our panelists some questions. I have a whole list of questions if I only hear crickets after this, so I want to make sure you all have an opportunity to ask your questions before I go into mine.
So Milton, I just asked our audience here to start to write out some questions and hand them over to me, so people are starting to do that. You might want to ask the Ann Arbor folks to do that. So you can keep asking your questions while I collect mine, and maybe Tim collects his over there, and then we'll start chiming in.
Hey, Andrew.
Yeah, Andrew, this is Michael. If while people are putting their questions together, I was wondering if I could just offer a quick response. I really appreciate Tim's comment, and I wonder, there's a design movement called Design for the Other 90%. I don't know if anyone's ever heard of that, but it makes the point that a lot of technological innovations benefit or service only about 10% of the world's population, and it's a movement intended to redirect the focus of innovation toward those developments that benefit the other 90%. And I wonder if, some of the concerns that Tim mentioned, do medical device manufacturers, entrepreneurs have an obligation to pursue developments that benefit the population broadly rather than those that focus solely on individual patients? Do we have some sort of ethical obligation to focus on the many rather than the few?
I think we're going to have a practical obligation because no one's going to pay for it. So in the end, CMS reimbursement, and it's still probably the best reimbursing country in the world, but CMS reimbursement, NICE reimbursement in the UK, whatever it is, if you cannot prove that you actually are reducing healthcare costs and doing better than today's technology, that is, you're getting better results for less money. So if you're getting minimally better results for 10 times more money, that technology is doomed. So we're going to have to focus on population-based treatments that have big impacts. That's one of the reasons ASI makes sense to me. If we can do a 20-minute outpatient procedure and prevent you from having to take 4 drugs a day for 30 years in a 20-minute outpatient procedure, that's a great example of the kind of thing I'm talking about. That is the only technology that will be economically viable in the next 20 years in the world, in the United States as well.
Well, but Tim, one second. I'd like to take your take as well as Mike as well. There are a number of healthcare systems, particularly in the state of California, that are forging ahead with making personalized medicine available to all of their patients and provide access to genomic-based clinical trials as those efforts become available. And what ultimately has happened is that it has allowed genomic testing covering some 40 conditions. And in addition to that, the hospitals are providing genetic counseling services to help patients make informed decisions about not only the outcomes of those tests, but changes or modifications in their daily behaviors and practices, as well as alternative treatment protocols or modalities that they should pursue. So I think that there is a broader effort, or should I say a marrying of healthcare services, both at the point of care physician or the GP all the way up to large hospital systems like Spectrum Health, that they are taking this personalized medicine approach on a much broader scale. And in that effort, I think we are talking about getting to population-based healthcare, or treating more effectively population-based healthcare challenges, for lack of a better word. Would you agree with that?
Yeah, I think, it has to be. It will ultimately have to be cost effective. I mean, they're forging ahead with it. It's new. They're depends on who's the best lobbyist. Mm-hmm. And historically, microbiologists have been extraordinary lobbyists for. We have identified the gene for disease X. We're only one step away from the cure. Yeah, that's understanding how the gene actually creates the disease. So, I mean, that's a 30 or 40-year process. So we've been hearing for 30 years, since I was a medical student, that we've identified the gene. We're so close to the cure for 30 or 40 years now, and it's probably another 30 or 40 or 50 years. And I'm just telling you, with the healthcare budget and what's happening in Medicare and CMS and Obamacare, we don't have 30 years to wait for these breakthroughs. I mean, I think these are brilliant things. I mean, everyone here is brilliant. These brilliant companies and brilliant diagnostics. These are great technologies. The question is, can you really impact population health and decrease healthcare costs while improving the quality and the length of life to a reasonable degree? We can't afford actually to have people live to be 100 years old. It's an interesting ethical thought as well.
Great. With that, I would like to open up to folks here, Gene, to folks in Ann Arbor to ask me questions that you have. They're being a little more process-oriented in Grand Rapids, no surprise, but I'd like to be a little bit more, a more open forum, if you will, with respect to any Q&A that you might have. Anyone here? Please, Dan.
I have a question related to business development. The presentations that you gave all came through pretty clearly to me in terms of what your value is and where you fit in the world. But my guess is you didn't start out that way. I'd be interested in any war stories you have on the difficulty of coming up with how to explain what you're doing to investors in the business world.
Excellent question. Before you guys answer that, were you able to hear that in Grand Rapids?
Go ahead and repeat it if you don't mind, Andrew.
Absolutely. Dan was asking about, from a business development standpoint, how you went from concept to where you are today. You didn't start out at this point. There was obviously a much earlier stage of concept creation and maturation that transpired.
And how to explain it.
And how did you explain it to the much larger investment and medical community as your technology evolved over time?
Well, I think we can all comment on that. In my experience, these things typically start with a vision, and maybe from drinking too much water. But you start with a vision, and usually the vision is captivating. You could describe a liquid biopsy. There's two words, and it gives you a vision. But where you go from there is you have to spend a lot of work at what would be the specifications of such a product. How do I define why it's better than what's available now? And that's true for all products. I mean, why should you use this rather than what's on the shelf? You typically spend a fair amount of time researching what the value proposition is going to be, what the specifications are, what the competitive advantage is going to be. those things don't usually start on a napkin. They take a fair amount of shoe leather is the best way to do it. Andy?
Yeah, I agree with David. It's not a vision, but eventually to be successful in the marketplace and to get money from, other people's hard-earned money put into your company, you have to show that you are going to be competitively the best for that sector. So, it's a lot of work first, identifying what is the competition, what is the current standard, and then how to make yourself better than what's available. And knowing that there are probably 50 other companies like yourself in the same field trying to do the same thing. So how do you rise up above the noise, always, making sure that you will have a competitive product? And that's how it should be. If you don't offer something better, you have no reason to be in business because, you're just going to waste somebody else's hard-earned money. So, it's a difficult, grueling process. You pretty much need a team that's committed to the vision that you start out, and then, you pretty much have to do the hard work to show that you are going to be better.
Brent, maybe you could comment from the Grand Rapids side, bring you into this conversation.
Sure, Andrew. I guess I don't have a lot to add because it's well covered, but I guess one thing I would say is that I've worked with probably well over 20 different surgeons and physicians that came to me with different ideas either on a napkin or they came to me just wondering, there's got to be a better way to do this. And they just said, where has the innovation been in this particular surgical procedure in years? Or I've been in the OR and I can't get out of the door in under 45 minutes, and this is such a simple procedure. So I think part of it is the practitioners, they see there's a problem, and they just know, and we start talking about the cost of healthcare, how much time do you spend in the OR, and how many patients can we get through, et cetera. So they're being driven by a need to do a better job, a more effective job, and that's the market driving them. And other ones are just very creative. And I'll just go back to what was said earlier is, from a technology side, we come up with different ideas and we prototype them, but then we surround ourselves with some very good people in the intellectual property space, some very good business and marketing people that help us understand the market as a whole and how we can differentiate ourselves and protect that differentiating aspect of our technology. So it's not a simple process and it's laborious, but you start. Sometimes it's opportunistic and sometimes it's strategic. That's what I got to add. Great.
Milton, have you collected some of those questions?
Yes, I do have two. I'll start with one. Just as an aside, I know that some people do have time constraints, so I think at 8 o'clock we'll call an end to the video portion, and then Grand Rapids and Ann Arbor can sort of do their own filling and talk with one another locally. So we have about 11 more minutes that we can ask questions. So the first one from here is, in information technology and in traditional manufacturing, new technology steadily lowers cost. This is often dramatically less true in healthcare, especially regarding medical devices. Can you explain why that is the case? Why is that happening?
Well, I think that's a great question, and it comes back to the point I keep driving home about healthcare costs and cost efficiencies and cost effectiveness, because we can put a price on a human life, and that's kind of the way that devices have been thought about historically. Okay, what is an acceptable cost? CMS will have as a formula, actually. Dollars spent per life saved or year added to a life, and it's where the human life's worth X. $20,000 a year, 30, whatever that number is. Choose a number. It's very arbitrary and ethicist. How do you put a number on that? You can't. And indeed, things like implantable defibrillators are very expensive, and on the average might prolong someone with a severe, injection fraction of 20%, dead heart, recurrent heart arrhythmias, and you get a defibrillator. It's a $40,000 procedure, and it might prolong your life a year. Okay, that's sort of where a bar is for medical device, but it's adding cost. Every time we add a year to that person's life, rather than letting them die, and use an expensive device to do it, we may prolong their life and yet we're increasing costs. So this is the challenge of medical devices. You have to have a device that can both prolong life, improve the quality of life, and do it at a cost that is cost neutral or cost saving to the way we practice today. I think we have to start thinking that way. We have to start thinking like IT does think, like the chips and everything. How do they keep improving efficiency of humans? That's how they reduce costs, and we are increasing costs.
Well, it's an interesting question because when you compare it to a manufacturing line, you manufacture a product and you sell a product. When you talk about extending a person's human life, obviously there's the quality of life, but how do you measure their productivity? I mean, so that's completely out of the formula, so it's really not a very good analogy directly. I mean, I understand what you're trying to achieve by using that, but how do you measure a person's value or their productivity? And I'll throw that at you, Michael.
Yeah, I don't know.
Exact number.
Yeah. To 6 decimal places, please.
$1,000 for quality.
Yeah. I think people refer to these problems as jagged edge problems because it's very hard to define a clear line.
Yeah.
How do you put a number at it? But we do have intuition sometimes that $100,000 for 10 more days of life may not be worth it, and you see that sort of thing sometimes. I think, no easy answers to that at all, and not a set of questions that we as a society have come to any kind of consensus on either, which I think makes it hard to move forward, but also even hard to talk about, hard to have a rational conversation about where do we set some of these limits.
Let me ask a possible switch on that, though. But if personalized medicine cannot reduce costs, but is there a way that personalized medicine could address another dilemma facing certainly both the biopharma and medical technology arenas universally, is the concept of patient adherence and compliance? Do you think that with personalized medicine that we're going to see better outcomes from patient adherence and compliance to a particular treatment protocol?
I think you have to clarify that question. I'm not sure what that means. I mean, medical compliance, and we have a big problem with drug compliance, not taking your statins, letting your prescription run out, not taking your antihypertensive in interventional cardiology, not taking your antiplatelet drugs and having a stent thrombosis and having a heart attack. So compliance is always an issue, and the cost of enforcing compliance, the best way to enforce compliance is education. It is the patient, the just good old patient-physician relationship. When I go into the patient's room after putting a stent in their left anterior descending, and I say to them, I'm prescribing Prasugrel Effient for you. You need to take this once a day. If you don't, your stent could clot and cause a heart attack. It probably triple the risk of clotting your stent. You know what? Those patients are very, very compliant when the physician has a relationship and spells it out that clearly. So I'm not sure that we have tools, Big Brother tools, diagnostic tools, blood test tools that are going to create compliance. I think we have to rely on education and physician-patient relationships, and that's the core of compliance.
All right. Milton, you said you had a second question?
Well, first a comment. I have a wife and daughter that wind up taking medication every day, and they're scrupulous about it. They beat me with sticks, and I still refuse to take my vitamins every day. And I'm semi-intelligent, so I fall into that category, Tim, of there are some people that are just so stupid, I'll use that word to describe me, they won't take the damn medication. So that's the dilemma. You've got to figure, I'm going to tell you, Brent, you've got to find a way to solve people like me.
You read the article that just came out, like New England Journal, showing that vitamins have absolutely no effect on human lifespan or any disease state virtually. So, I mean,
okay.
So actually, you're actually way smarter than you think.
I am so going to have my wife talk to you soon. Okay. So here's the question. It's another ethical question. So the first part that I'll ask is how many hospitals across the state have an ethicist on staff? Just out of curiosity, a percentage. Is it 1% or is it 98%? Just out of curiosity. But the bigger question is, in terms of what you do you guide patients and clinicians to answers, or do you help them to develop or know the right questions? So what exactly is it that you do?
Yeah, questions. I think there's probably about 5 or 6 of us in Michigan who are paid to do this. So not too many, but usually you find paid ethicists at the larger medical centers, academic medical centers. Every hospital has, or almost every hospital has an ethics committee, which is something that's mandated by the Joint Commission as a mechanism for addressing or resolving ethical challenges that come up in patient care. So you always have some sort of ethics infrastructure in a hospital. The way that we understand ethics consultation with our ethics committee is we make recommendations and we offer guidance, but we're not authorized to make decisions at all. Clinicians oftentimes, I think, are looking for somebody to make a decision. If they have a tough scenario and they want to know, should I or shouldn't I, they often want you to come in and say, do this, and write it in the chart and be done with it. And we try to resist that if we can, and see our role as, like you suggested, asking the right questions, trying to highlight what are the pros and cons associated with one option or the other. People who are authorized to make a decision can make an informed decision, an informed ethical decision. Just like we expect or ask patients to make informed medical decisions, we want to make informed ethical decisions.
Great. Well, Milton, because of time, you mentioned about wanting to stop at 8 o'clock. So first, let me thank everybody in Grand Rapids for attending tonight, and everyone here in Ann Arbor, thank you very much as well. I want to send a special thank you to Tim and Brent, Michael for participating, as well as Kalyan and David on our side here for rounding out this panel. Of course, if you have any questions beyond tonight, please feel free to email them to us, and would like to thank Milton as well for bringing this vision to fruition. So thank you very much, all of you.
Before we turn off the camera, thank everyone again for coming out. We'll continue to email you. We have your email addresses. Our next appearance will be at ACE. We'll be presenting the Technology Review Top 10 Breakthrough Technologies, and we have a panel of Michigan's new technology specialists that'll be appearing in that panel with us. We'll be February 19th, if I remember correctly. We're doing microbreweries. We'll be hosting that in Grand Rapids, in Traverse City, in Southeast Michigan. Same forum. What's the technology behind it, and what's the taste? And then the one after that, I think, is March 19th. We'll have an astronaut here in Michigan with 3 degrees from MIT talking about the views from outer space and what that means to our educational system. So stay tuned. We have some really exciting programs. If you have ideas, please email them in to us, and we're always looking for volunteers. Let me repeat that. We're looking for volunteers, so please, come out and help us. My thanks again to Menlo Innovations. My thanks to GR Current for hosting us, and we're going to sign off the air. Thanks, folks. Drive safe, please.
Okay.